So welcome everyone. Welcome to the first colloquium for the semester. I hope you're all doing fine. It's a pleasure to have Archishman Raju speaking to us today. So Archishman did his PhD from Cornell, where he worked on studying renormalization group theory and applying it to information theory and so on. He was in a postdoc in the Rockefeller University. He was an independent postdoc there. We worked on geometric models for trying to understand how cells get their fates specified. So from the past 7-8 months, he's at NCBS and the National Center for Biological Sciences, and he's interested in the mathematical modeling of development and evolution. So today he'll tell us about how to go from microscopics to phenomenology and how to think about the Waddington landscape in specifying cell fate. So if you have questions, you can either raise your hand or maybe try to unmute yourself and ask questions right away. So Archishman, the floor is yours.
Okay, thank you, Vijay. My audio is okay?
Yeah, perfect.
Okay, great. So I'm going to talk about Waddington's landscape, and hopefully as I talk, my choice of title will become clear. And the work that I'll be talking about is all based on, most of it is in this paper that's referenced, and that includes all of the collaborators that I have worked with on this. So the outline of the talk is that I'm going to just give a very brief introduction to development and systems biology models, particularly for those who might not have seen developmental biology before. Very, very quick introduction. And then I'm going to tell you about Waddington landscapes and what their relevance is and so on. And then most of the talk I'm going to focus on thinking about how you can use the metaphor of a Waddington landscape to mathematically model systems in developmental biology. And I'll go through several examples, in different model systems and different organisms at the end. Okay, so a very quick kind of introduction, which is not just to get a sense of the concepts that one is dealing with. In development, you often talk about you talk about the fate of the cell. And so at the most basic level, you start as a single cell, and then you develop into a whole organism, and the cells divide, and each one of them end up taking a particular fate. For example, a skin cell is different from a neuron. And the question is, how do cells acquire this particular fate, starting from a single cell where nothing is specified. And the way early embryologists would talk about this is they would draw what was called a fate map. This is sometimes still used. And the idea behind it was that you take an early embryo and you mark sort of different regions of that embryo. And then as time progresses, you can see what happens to those regions and as the organism develops. And so this region here, for example, might become the eye. This region here might become some segment in the larva. And the cell fate was said to be specified or actually determined once these regions had, you could not change what fate this cell was going to take. And so in particular, there are many kind of transplant experiments that were done where you could take a region here and put it somewhere else in the embryo and then see what happens to it. And the question was, if I take this region and put it somewhere else, will it become a segment or will it become an eye? And depending on what its fate was, whether it became a segment or an eye, the cell was said to be potentiated or it was said to be determined. And so cell fate specification is really asking, how does one go from a point where the cell hasn't decided its fate to how the cell reaches a point where its fate is specified. And of course, in complex organisms, the cells move around, and so actually these fate maps are not totally accurate. But in some organisms, in particular in C. elegans, in the worm, which is somewhat special example, but that is an example where you can really track each cell. It's about 1,000 cells, and you can really track each cell and see what kind of fate it'll end up taking. And the important thing from the point of view of the talk is that, so this is this figure, which the details are not at all important, but the important thing is that there's some sort of hierarchical structure in terms of how the cells get specified. And this sort of structure, is important when one thinks about how to model cell fate specification, as I will make clear. So then the next question is, how does one model the system? So how does one model what's happening to the cell when it's taking a decision of whether to become an eye or a segment? And the usual way that one would think of modeling this at the level of the cell is actually quite complicated. And in some ways one could argue that what's happening inside the cell is even more complicated than what's happening in the embryo. And in the cell, what's happening is that you have some external, one of the ways is you have some external inducing signal, often called a morphogen. And, so it's some ligand, which, the cell has some receptors, and the ligand binds to the receptors. And then there's some complex gene regulatory network inside the cell, which, what's the question? Oh, sorry, I heard a noise. So there's some complex gene regulatory network inside the cell, and the gene regulatory network then, which these are all proteins which either activate or repress other genes, and there's some transcription. I mean, so there's some complex network of interactions, which then finally leads the cell to make a decision. This is one example. It's from rat cells, which have to decide whether they want to differentiate or whether they want to divide. And the decision in the end usually depends on the concentration of some final protein. In this case, it's called ERK. And the way that one could think of modeling what's happening inside the cell is to really try and write down differential equations for each one of these components that one knows are involved, and that are involved in this regulatory network. And so if one tried to make a model like that, that's written on the right. And again, the fact that you can't read it is on purpose to show sort of how well, what these models are like. But if you zoom into one of these equations, then you see that sort of the concentration of these different chemicals inside the cell are given by these classic Michaelis-Menten type of equations. And all of these, as you can see, have some parameters in them. There's some rate constants, some thresholds, and these parameters are usually not measured, and it's usually too much time to measure them or to really care to measure them. And but the predictions of a model like this, of course, depend on these various parameters in the model. And so these kinds of models are quite over parameterized. And in fact, one can sort of precisely using information theory say that these models are sloppy, in the sense that most directions in parameter space for these models don't really matter for the kinds of predictions that you're interested in, which can, for example, be the level of this ERK protein. But this is not immediately obvious at the level of the model, because changing a particular rate constant here might actually have quite a significant impact on the concentration of the ERK protein here. So this is just to say that gene regulatory networks and that way of modeling how a cell is taking a decision. Hello? Or maybe some. Is quite a complex and sort of a sloppy way to model the system. Yeah.
So here, this what you have here is a reflection of the number of equations roughly that would be needed. So it's a set of coupled equation, 20 equations or 30
right.
equations or something like that. Is that the generic kind of
Right, and the number of parameters is sort of quite large.
Right,
each one of these
right.
equations have, in this case, about 6 parameters. And actually, so this is, I mean, this is sort of what I am here calling a microscopic model. Of course, it's not microscopic in the sense that it in any way accurately captures what's happening inside the cell. which is more complicated than this. But for my purposes, this is sort of what a kind of microscopic model you could make of how the cell is taking a particular fate.
And so each of the variables, I mean, forgetting about the parameters, which, as you said, are some rate constants and thresholds, but each of the variables in these differential equations, these are just these various chemicals that appear in your network there.
Yes. Correct. Yes.
So there are about 20 or 30 of these chemicals which are relevant that you have to kind of keep track of. That's. I just wanted to get a sense for. I mean, all 20 or 30 of these are crucial to the.
Right. Right. No, so that's a good question. I mean, and this is, I mean, the fact is that they're actually not crucial. In fact, there's some perturbations you can make to the system blocking some of these things, which doesn't change the system behavior so much. But then there are other perturbations which can change it completely. So it's not obvious how one should identify which are crucial and which are not. And this is in general a difficult question. Yeah, but it's not right. It's not clear that all of these variables are absolutely important if you're interested in a particular decision that's being taken by the cell.
Yeah, I mean, it sounds a little like, I mean, the kind of equations they are like in a renormalization group flow where you have a large number of relevant and irrelevant directions, I guess, and you want to find out which is the which are the relevant, whatever the critical surface and the off critical surfaces. So yeah.
Right. No, exactly. And this is the sort of, I think, this sort of thing is what one wants to do for biology, which I don't think really a procedure exists for, and for various reasons for that. One being that in physics you have some symmetries, you have some way of writing down terms, whereas in biology you really don't have. Anyway, so this will sort of distract but yeah, this, I agree, this is a good point, and this is something that I think is really the goal of what has to be done here.
Okay, thanks.
Yeah. So the alternative approach, which actually, I mean, it's the renormalization group was brought up, and I think actually came from sort of different kind of philosophical difference from a gene-centric approach to cell fate specification. And well, I think one could argue was really an early example of talking about emergence and emergent descriptions in science. Was done again by, you can see in 1957 by Waddington, who was also an embryologist. And the way he conceptualized the cell taking a decision at the level of metaphor, and this actually picture has become quite famous now, and is that he thought of it as a ball rolling down a landscape. And the different endpoints in this landscape corresponded to the different fates that the cell could take. And the important thing about this landscape, there are several different important things that he pointed out about the structure of a landscape like this. One of them was that this landscape was what he called canalized, which means that you can see it's sort of in the form of these different canals. But what he meant was that really the cell was quite robust to perturbations. So there are different decisions that the cell could take. But once it went along a particular trajectory, so if this was in one of these canals, then it was very difficult to perturb it from that trajectory. So there were some decision-making points where the cell was sensitive to perturbations, but then there was a lot of the trajectory where actually the cell was not very sensitive. So this was one thing that he was trying to capture, which he used to explain the fact that actually development is very robust. You don't actually end up getting very strange things usually. And it's quite remarkable that despite all the noise, despite all the difference and initial conditions one could imagine, you robustly end up developing an organism. And the second thing was that you don't usually end up getting new cell types so easily. And so if you make perturbations in this system, for example, you knock out some gene function or you put in some chemical from outside, what happens is not that you generate a new cell type. What happens is that you permute between known cell types in the organism. So this was sort of an important insight that the dynamics of this system doesn't small perturbations don't actually qualitatively change the dynamics of the system, but they really change, if you have different cells in this landscape, they sort of change which of these minima they would end up in. They don't create new minima. And here by perturbations, I mean external perturbations that you could do in an experiment, like, giving a heat shock, putting ether, or knocking out a gene, or whatever, things like that. And so this idea of a landscape was a very nice metaphor, but it's sort of lost it well, it's lost in popularity for various historical and sociological reasons. But it has recently been revived, particularly with interest in stem cells, and the idea that uh, again, cells can be potentiated to take different fates. So this metaphor has become quite a useful one to think about sort of new experiments in biology. But this metaphor, one can ask how it can be converted into a formal description which can actually be used to model what's happening to cells, rather than simply give an intuitive explanation of what is going on. And so that's going to be sort of the main part of my talk. And so in contemporary times, there has been some, revival and thinking about geometric approaches to development and cell fate specification, which are sort of agnostic to these complicated regulatory networks, which, as I said, in any case, parameters of which are not measured and which one doesn't know fully. But they realize that the at the phenotypic level, at the fates that the cell is going to take, the system has has a somewhat simple nature. There are a few cell types, and you permute between those cell types, and there's sort of a hierarchical nature to these kinds of decisions that are taking place. So the question is, how does one mathematically model this? And
I have a question actually.
Yes.
Can I ask a question?
Yes.
These equations that you showed,
Yes.
they are all first order in time in some sense.
Yes.
So why would it be that there is no? Why are they like overdamped Newton's equations? Why isn't there a second order term? That's the first question. The second question is, why isn't there noise terms?
Right. So noise terms are there. They're just not put in there.
Okay.
But certainly there's noise. In fact, there's number noise, and that number noise you can actually write as proper noise terms in these equations. And they're first order just in the sense of their chemical kinetics. So they're already approximations. These are already phenomenological equations. They're not meant to capture the real microscopics. Yes.
Okay. Thank you.
Right. So this thing of Waddington landscapes, it turns out the kinds of characteristics that one wants in development sort of correspond to what in dynamical systems are called Morse-Smale systems. And the idea in Morse-Smale systems, I won't go into the details of the mathematics, but is that if you have some vector field and you impose some reasonable conditions on the vector field, and for example, you don't want chaos, and you really want some finite number of periodic orbits, finite number of fixed points, and then you need some conditions on the insets and outsets. But I won't go into the details, but the idea is that conditions which seem quite reasonable for developmental systems, where there is some notion of fixed points and going to some finite number of fixed points, you can always write such systems as a potential, as a gradient flow. And it turns, so this is some work by Smale in the early 60s, and it turns out that you can't quite write them as a gradient flow, but you also need a metric. And so vector fields of the kind one is interested in development can be written as a gradient of a potential times a metric. And so this is sort of the first insight which formalizes the idea of a Waddington landscape by saying that you can in fact write these dynamical systems. You can take all these first order equations, for example, and you can write them as a potential flow, but you have to include a metric to really, if you want to fully capture the vector field. And so just to give an example, you can take a simple system, which actually is an example that I'll use later on, but you take some bistable system, which has two stable fixed points and a saddle point in between. And the vector field that you have is shown here in green. So it's some, some vector field which is kind of strongly compressed along the vertical direction, and then it flows to one or the other fixed point. And now you can, you want to write this system as gradient flow. So you write down some potential, bistable potential, which is of course easy to write. And the potential, some generic potential that you will write down won't correspond to these, the flow of that corresponding to that potential, the gradient of that potential won't of course correspond to these vector field lines. But what you can do using the metric, so the contours of the potential are drawn here, and the normal to the contours is, is shown in red. And what you can do is you can rotate the normal to the potential and make it actually align with the vector field that you're interested in. And this is the job of the metric. So
where do you get the metric from? From what principles?
Well, so this is, this is a good question. In this case, one can, come up with some construction for this metric using sort of a sum of projection operators. But here I'm just taking an example where the vector field is given, and then you write down some bistable potential, and then given that bistable potential, you try to align locally at every point. You try to align your vector field with the gradient of the potential. This is not something that one would actually want to do in practice, but this is just to give an example of what the potential is doing and what the metric is doing. In particular, the potential is storing the location of the fixed points, right? You want the location of the fixed points and the saddle points to be in the potential, but it is not storing all of the dynamics. And so for the dynamics to correspond, you need the metric. And so there is this sort of division where, given some arbitrary dynamical systems, you want all the fixed points to be in the potential. And you then want the metric to give you the right flow at every point. So this is how, any arbitrary vector field one could write, not arbitrary, given the conditions that I was talking about, one can write as a potential times the metric.
But so there is no biology information going into the metric, right? For example, in conformal field theories in 2 dimensions, we encounter these equations, and the metric actually near the fixed point is fixed by, what's called as a Molotchikov metric in terms of, correlation function of 2-point functions. So I'm just trying to understand where is the information in biology for this metric? Right.
No, it's an interesting question, which, so the metric, so this is sort of one has to be a little, how should I say? There are less principled ways to do this in biology, but the information that you want is that the flow, so at the first step, what you could do is that the flow near the fixed point should be correct. And so if you take you take a linearization around these fixed points, then that linearization around the fixed point, is some matrix, which in general is not symmetric. And you want that matrix to be written as the product of 2 symmetric matrices, one of them being symmetric and one of them being the gradient. So if one had some access to the flow near these fixed points, then that would be one way you could fix them near the fixed points. And then interpolating between the fixed points from a biological perspective is sort of trivial, because you don't have that kind of information about the dynamics in any in biology. You don't have that kind of data.
Okay. Thanks.
Okay, so the next thing that you want to do, right? So you have this procedure. You can have a landscape, and you have some way to write down a potential. But the next thing you want to do from the perspective of the biology is you want to parameterize the landscape. As I said, you can do experiments where you knock out genes, so you put in some external chemicals, and when that happens, you permute between different known cell types. And so you need some way to parameterize the landscape and have it give, different. So in particular, you need some way, starting from some system, for example, which is tri-stable, to give a simple example, you need some way to go to a landscape which is like this. So that if, for example, if a certain perturbation only gives you cell types which correspond to the central attractor, then that perturbation, corresponds to some parameterized way to go from this center diagram to the diagram on the left. And similarly, all of these are just enumerations of what you could get for a tri-stable landscape, either the left or the right attractor, and or only one of them being present. And so what you do then is you use these you associate these attractors with sort of cell fates that you know are there in your system. And so I'll go into more detail in this in the examples. And then you want some way to parameterize them and see, given some external experimental thing that you're doing, which typically is sort of induction experiments where you overexpress some protein, or genetic knockout experiments, you want some way to change the landscape to parameterize it. And of course, naturally, what here I'm talking about are bifurcations, and so you need to parameterize the landscape and see what bifurcations can take place. And it turns out that there are really only two bifurcations that you need to worry about. And the most important one really is the saddle node bifurcation, which is that you have an attractor and a saddle, and they annihilate each other, so leaving you with nothing. Or conversely, as you change a parameter, you get the appearance of a saddle and a stable fixed point. And the reason for this, the reason one can impose something like this is through this thing called genericity. And it's sort of an interesting difference, again, I think, from physics, where one is used to having symmetries. And then writing down some Hamiltonians is consistent with the symmetries of the system one is interested in. In biology, particularly in the kind of system that I'm dealing with, that we're dealing with here, is gene regulatory networks. There's no particular expectation of any symmetry, but there is some expectation of genericity. And again, the mathematical definition is sort of technical, but the sort of simple idea is that if I perturb the system, if I put an arbitrary perturbation on my system, then that should not change the qualitative behavior of the system. And so if I have a system with a saddle node bifurcation and I put some arbitrary perturbation on it, small perturbation, then I should still get a system with a saddle node bifurcation. And it turns out for other kinds of bifurcations, usually they require, for example, pitchfork bifurcation is a common example. They require some symmetry in the equations. And small perturbations actually break up those bifurcations and convert them into a sets of saddle node bifurcations. And so this kind of imposition of genericity is quite powerful because it allows you to then talk about what kinds of behaviors you could get as I change parameters. Again, here first, just to point out, so far I made no reference to the actual equations. This is again sort of generically from dynamical systems. But this condition sort of allows you to really classify different kinds of behaviors, different kinds of ways that my landscape could change. And the heteroclinic flip is also important from a biological perspective. What it really says is, so if I am at this attractor A at the top and I have a saddle node bifurcation with this saddle small a, then the question is, where will I flow to? Will I flow to C or will I flow to B? And that's something that you can change by changing the position of the saddle. So it's more of a global bifurcation, but it sort of captures something that's very important to biology, which is that if a cell state loses its stability, if I go down the developmental landscape, where am I going to flow to? Which is quite essential, for the kinds of things that we're dealing with. So I'll pause after this, but just to say that it turns out using this condition of genericity, one can write down what I'm calling phase diagrams, but this is very vulgar use of the word, not to be taken seriously, but one can really classify the different kinds of bifurcations that can take place. And what I'm drawing here is that you, as I said, so forget the heteroclinic flip, you're interested in the saddle node bifurcations. And the saddle node bifurcations, so let's say you take systems which have three stable fixed points, and you have two parameters that you can vary. There's a low dimensional system, but it's actually quite useful for biology where you're often dealing with such low dimensional systems because there's this hierarchy in developmental decisions. And so you have such a low dimensional system, you have two parameters you can vary, which is again typical. You have some two proteins whose concentrations you can change. And you have these three known cell types that your system is going to permute between. Then in that two parameter space, there's a line of saddle node bifurcations, which corresponds to losing one of the attractors. And then you just have to work out how these different lines of saddle node bifurcations meet. And there are two ways they can meet. They can either meet in a parallel way, forming what's called a cusp, or they can meet transversely. And so it's it's not too difficult to just work out the different ways in which you could really transition from a three attractor system to to bistable and monostable systems. And so you can see, for example, in this example of what's called a dual cusp, you start with a tristable system where all three fates are possible, and then you undergo a saddle node bifurcation on the left, and you lose the central attractor. And so now you just have the left and right attractor. And then if you go down in the yellow line, then you use the right attractor and you just have the left attractor. So this offers sort of a minimal parameterization of the possible behavior in your system. I haven't told you how to parameterize this. I'm going to do that in the examples. But these diagrams just follow from this imposition of genericity and some rules for how these different saddle node bifurcation lines can meet each other. So maybe I'll just pause here in case there are questions. After this, I'm going to go into the examples.
Yeah, so maybe just to connect it with what you started with, where you had maybe 20 or 30 different chemicals and many parameters. Is it so at some point I had the impression you were trying to understand the qualitative features of the landscape of that system, or are you further specializing, like in the case in your slide right now with just a few, 2 or 3 because high dimensional parameterizations of these flows are likely to have qualitatively very different features from low dimensional ones. So which sort of regime are you in?
Right. No, so I mean, this is sort of the interesting and I think essential question, because the high dimensional parameterization was again at the level of what's going on inside the cell. But at the level of what the cell is doing, you have sort of, again, a low dimensional set of things that the cell does. So in that example that I had mentioned, actually there are only 2 different qualitative behaviors that the cell makes. Either it divides, depending on the level of this protein. Either it divides or it decides to differentiate into a neuron-like cell. And so there is this high dimensional parameterization depending on the external signal, but the output is sort of low dimensional. And of course, now you can argue, well, why then the high dimensional parameterization? And that's because, of course, there are other things that the cell needs to do. But the claim I'm making here is that to try to model the output for a particular decision that you're interested in studying using the high dimensional parameterization is sort of a sloppy way to model it, when actually there is a more there's a lower parameterization which at least captures a certain subset of what the cell can do, which is what you're interested in a particular experiment. So in that sense, I'm trying to argue that it's a more principled way to treat biological experiments rather than and a more phenomenological way. You don't quite know what's going on at the level of the cell, but you capture the kinds of observations you're interested in.
Yeah. Anyway, the earlier one was also, as you said, phenomenological, and perhaps in spirit, you're just trying to capture the, so to say, the relevant directions. I mean, just a few directions in that space, but you can't do that explicitly, but you can try to do it phenomenologically.
Right. Mm-hmm. Exactly. Yeah. And it's quite interesting. I mean, how this high dimensional crazy system reduces onto this low dimensional manifold is quite a I mean, this is quite, and what consequences that has for how you would want to model is quite interesting, I think.
You really don't know how to do that, right? I mean, you have these set of equations, and so there is a landscape in which there are flows, and there are saddle points and all types of things.
Right. So from
from there, so I didn't have discussed that to discuss what you're discussing now, because I don't see the connection very easily. Or you see, there must be, you're saying.
No, there must be, and I'm not claiming to make the connection in this, but here I'm using the biology to say that, well, in fact, qualitatively, actually, there's some simple description that's possible, which was this thing of Waddington landscapes. And so I'm going to not make the connection, but use that qualitative description and then say, without making any reference to equations, how much can I get just by general considerations, without knowing about the particular flows? And so the biology is important in the sense of these observations that Waddington made, that when you perturb the system, you don't, fundamentally shift the landscape. You just have the same sort of system, very similar system. And right, and second, that there is some downhill flow where these cells are getting specified. The fate is
In some sense, you're trying to do some sort of a topological classification, right?
Yeah, correct.
Because
Yes.
most probably you'll talk about this hopefully, Morse
Right.
index and stuff like that.
Right, correct. Yeah. So this is really, I mean, essentially, it is really topology, because that's the relevant mathematics, correct? Yeah.
Okay.
Yeah.
Thank you.
But actually, so I'm not going to go into the mathematics, but I'm going to shift and talk about the examples and how this is relevant to um, can be actually relevant to modeling fitting biological data and so on.
Maybe you'll say this later. In the earlier case, it was sort of evident what the independent variables are, like some chemical concentrations and all.
Right.
In this coarse grain picture, what are the things what do I measure? What are the...
Correct. Yeah. Right. Yeah, so here these axes one doesn't know. And again, this is the thing, that this is some low dimensional projection of some high dimensional space. But what the way one makes sense of it is by saying that these different attractors correspond to different fates that the cell can take. And so that's how one makes an association with the experiment. But what these axes are these are abstract axes. Yeah.
Okay.
Yeah. So maybe I'll go into the examples to see if in the first example actually is from C. elegans, and there it's I'll just go through it quickly because it's actually from work that was done in the past, where as I said, there are some in C. elegans you can really say what's happening to every cell. And so at a particular stage of the development of the vulva in C. elegans, which turns out to be a well-studied system experimentally, you have these 6 cells. You can ignore this one. And there are 3 different fates that they can take, which have some names that have been given to them. And you know the pathways that are involved in deciding what fate the cell takes. So it turns out something called Notch delta pathway is involved, through which the cells communicate with each other, and some protein called epidermal growth factors involved, which something called, there's another cell which sends signals to each one of these cells, and there's a gradient depending on whether they're at the center or at the extremes. And so these two signals together make the cells decide which fate they're going to take. Now again, so you want some 2-dimensional landscape, which is 3 attractors, and you want to fit experiments which will change the level of the EGF. Many such experiments have been done, or change the level of the notch, notch delta, knock out the notch delta pathway, and depending on that, the fates that these cells take changes. So in the normal, what's called the wild type, this takes red, this takes green, this takes blue, but depending on how you change one or the other signal, this can become all red, or there are different kinds of permutations of red, green, and blue that these cells will end up taking. And so it turns out, so then this example is just to say, well, how does one then parameterize this system, again, without going into the details of the pathway and how EGF and notch actually work inside the cell. How does one parameterize the system? And so you want 3 attractors. So you want some potential which has 3 attractors, and you want the parameters in your potential to be able to change which of these attractors are stable. And then you put some noise in the system, and then you try to fit experiments where depending on different perturbations, and so the EGF and notch perturbations you map onto your parameters, and depending on different perturbations, your landscape has a particular structure, and then the cells end up taking different fates in different proportions. Just to go through very quickly, just to give the setting of how this can become useful as a tool to fit experiments. But I'll just move on to another example, which sort of I'm working on now, which is in the mouse blastocyst. And so mouse blastocyst is very early stages of the mouse embryo. In the mouse embryo, so this is about 4 days in the mouse embryo. So it turns out initially you start with a single cell, something called cleavage happens, and you get these things called blastomers. And after that, there are 2 decisions that are taken. So there are 2 choices that these blastomers have to make. At the first stage, they adopt a fate that's called the trophectoderm, the inner cell mass. And the second stage, they take something called the primitive endoderm and the epiblast. And the second choice that I'm interested in studying, again, these are things that sort of have been experimentally studied a lot, because actually it's quite an easy system. This is before the embryo gets implanted um, into the uterus, and so it's easy to sort of take it out and change signals and still see the behavior of these different cells and what kind of fates they end up taking. And the epiblast actually ends up becoming the embryo proper, and these are just supporting tissues. But in any case, this is sort of an early decision that cells have to take in the mouse embryo. And then you can ask again, how do they take this decision? And the way that this is modeled, so to relate actually some of the questions that were being raised, the way that this is modeled is to again say, and this is again a very actually a small, comparatively low dimensional model of what's going on inside the cell. But there are some genes that are involved, GATA 6 nanog. There's some the cells communicate with each other using this protein called FGF. And then depending on the level of the FGF, the cell decides whether to become a primitive endoderm and epiblast. And usually that depends on which gene is being more expressed, or the GATA6 or nanog. That's how they decide what fate it is. That's a marker for the fate. And the kinds of experiments that are done here are to put in a lot of FGF or to knock out the FGF receptor, and then to see again what kinds of, how does that change whether the cells become primitive endoderm or epiblast, to get a sense of the developmental decisions and what they depend on. And so I haven't written them here, but you can imagine a model like this, which it does in fact has equations for each one of these components, for GATA6, for ERK, nanog, and the FGF. And then based on those equations, it tries to fit these experiments and find some agreement with what happens when you change the level of the FGF. But again, the argument that one is making is that this is far too high dimensional representation of what's going on, given the kind of experiment that one is interested in. And in fact, you can take this model and you can make a two-dimensional sort of cross section of it, which I've done here. And it turns out that this model is, the essential behavior of this model is identical to one of the diagrams that I showed called the dual cusp. And so doing this actually gives you a much more intuitive sense, first of all, for what is happening, because you can have the FGF on one axis and you can have some other parameter on this axis, and so you have some two parameters, and this diagram really tells you what region of the landscape you're in, whether you just have the primitive endoderm being stable or the epiblast being stable. And the kinds of, so what the model does is it tries to fit, if I put in some external FGF, what happens to the fates of the cell. Turns out they all become primitive endoderm. But the experiments being done, are putting in a lot of FGF. They're really saturating the system and then seeing what happens to the cell fate. So what this kind of picture does is also it allows you to really get a much more sensitive, dynamical picture of what's going on. And it's obvious, for example, that here what you're really interested in is not really putting in a lot of FGF, but titrating the FGF, and then you can really get a sense of how the FGF pathway controls the decisions in this system. And second thing that it does is, this dual cusp can be parameterized again in a minimal way using the kind of polynomial I showed. And there the parameters again now very directly correspond to the kinds of decisions the cell is taking. Whereas in a systems biology model, it's very unclear how different parameters in the model actually relate to the decisions that the cell is taking, because again, it's very high dimensional representation of what's going on. The third thing which I think is interesting is related again to the questions is, it turns out that this model is actually very near this thing called a dual cusp, which is a codimension 2 bifurcation. But then this means, of course, this is basically it's near a critical point. And this should allow one to really write down, and something that was discussed here, allow one to go from the high dimensional system to this low dimensional one in some principled way, where most of the dimensions are really compressing, and then the relevant dynamics is taking place in this low dimensional system. So this is sort of an example of how one, I think, learns sort of more, gets a more intuitive picture of what's going on in these experiments, but also, it sort of tells you a little bit more of how you should, parameterize it, but what kind of experiments will give you more information about the system. So actually, I think I'm about 42 minutes in. So maybe I'll quickly go through the second part of my talk, which um, so the second part of my talk actually was going to go in. So this I was giving you an example of these low dimensional spaces where you have a few cell fates and you're interested in how external signals are permuting between these few cell fates. And sort of I showed you how without any reference to the dynamics at all, which in any case is hard to get access to, you can still make nice parameterizations for how cells will take these different cell fates, and these follow just from these constraints of genericity. And the second part of my talk, I actually want to talk about spatial patterns. And so here, so far, I wasn't really discussing space, but of course, space is something that you're interested in when you're, when you're talking about development. You're really interested in how different cells at different positions take different fates. How does something at your leg decide to take a different particular fate versus your eye, corresponding to the picture I showed at the very beginning. And so the sort of paradigm, so there are two ways of doing this. So two classic ways that developmental biologists talk about this. One is called positional information, where you have an external morphogen, and depending on the level of the external morphogen, which varies as a function of position, your cell decides to take a particular fate. The second way, which is usually called self-organized pattern formation, comes from this Turing mechanism, and which I'll go into the next slide. But the Turing mechanism is some way to capture these patterns qualitatively in a very nice way. And so here are some examples from seashells and fish, where on the left is the actual system, on the right is the computer simulation. And you can see that you're able to capture these spatial patterns very nicely using these computer simulations of relatively simple systems. And the kinds of systems that you're dealing with, I'll show you. So for example, this is something, classic model is called the Gierer-Meinhardt model. And what you have is some partial differential equation, which has two things. It's usually an activator and an inhibitor. And the activator activates itself, it activates the inhibitor using these terms, and the inhibitor inhibits the production of the activator. And the reason that one would be, so this is already a very nice way of talking about spatial pattern formation. It's obviously some very coarse grained model to begin with, but the reason one would want to think about this geometrically is, again, one cannot take the analogy of the microscopics too easily. So in fact, it's very difficult to find any biological system where you have an actual activator and an inhibitor which is creating a spatial pattern. So qualitatively you can make an easy match, but then one cannot take the microscopics too seriously and try and really find activators and inhibitors inside which are generating these patterns. At least so far, I would say the search hasn't been very successful. And so that's why, this was one thing that we also wanted to look at biologically, geometrically, sorry, to see if sort of it gives you more of an insight into what these Turing models are doing. And the behavior of this model, so the very simplest level at one dimension is just there's a formation of a bump, which is this pattern state, which you're going to call the pattern state. And so you have an homogeneous fixed point to begin with, which is unstable to diffusion. And then when you increase diffusion, and diffusion of the inhibitor needs to be more than the activator, you get some pattern state which leads to this bump, both in the activator and the inhibitor. So it's a very simple model, and sort of in one dimensional, this is periodic boundary conditions. There's not much that it can do. The second thing, sorry, I should quickly mention is that these equations, of course, are arbitrary. These people come up with some terms here for, but again, these are completely, in fact, I've written terms which do not correspond to the usual model, but change them slightly. So that's another sort of, okay, so that this model forms a bump, and now you can ask, well, can I write down a potential which gives me, something that goes from a homogeneous state and gives you a bump at the end? And it turns out that in the way the model is formulated, it's quite hard to write down a potential. The model is not obviously potential. In particular, the cross terms actually are not equal. And in fact, you can show that it's quite difficult to write down a local potential of two fields, where one acts as an activator and one acts as an inhibitor. So directly trying to write down, some gradient flow for the kind of partial differential equation that I showed actually is not a very useful direction to pursue. So that sort of made us think instead of looking at the discrete case, because the continuous case seemed a little puzzling. And in the discrete case, so let's just say you take the simple example, you just have two cells, you had some continuous dimension, now you just to make two discrete cells. You can ask what does patterning, what does a bump correspond to here? So it just corresponds to one of the cells being high in the activator variable and the other cell being low. And which cell becomes high and which cell becomes low can vary. That's sort of basically a symmetry in the system. And depending on the initial conditions, one of them becomes high, one of them becomes low. And here, sort of the geometry is pretty clear. It's actually 4-dimensional. I'm just showing you 2 dimensions, A1 and A2, and you have some homogeneous homogeneous saddle point, and then you start near the saddle, and then you either flow towards this fixed point or you flow towards this fixed point. So geometry of what is happening is pretty clear. Now you can take something, a little bit, you can look at 5 cells with 5 different stable fixed points. Again, they're just permutations of which cells are high and which cells are low. And you can look at the linearization around the homogeneous homogeneous saddle, where all the cells are the same activator and inhibitor variables. And turns out the eigenvalue spectrum there has only a very few eigenvalues, in this case 2 eigenvalues, which are unstable, and most of them are stable. So the unstable manifold of this homogeneous fixed point, which is leading you to the pattern state, is actually quite low dimensional. So even though you're modeling this in, this is a 10-dimensional system in the discrete case, your unstable manifold, which is the relevant dynamics. Now this is in space, so this one should not confuse with the chemical system. But in space, the unstable manifold is actually in Turing, it's quite a low dimensional system. So this allows you to actually write the system as a potential.
I have a small question, if
yes.
you don't mind.
Yes.
Just in the previous slide,
yeah.
See, what I do not understand is, okay, there is a unstable manifold of 2 dimensions.
Yeah.
And so you linearize the equations around the fixed points.
Yeah.
How do you fix the parameters which tell you that there are so many unstable directions and so many flat or so many stable? I mean, is it experimentally done, or how do you know this?
Right. No, so this is very, this is because the you don't know this in the sense of you can't really fix these parameters. These parameters that I've written here, there's no real way to fix them. As I said, you don't even know what the activator and inhibitor is. You can try to make some qualitative agreement, and you can fit it to these parameters, and that's one way of doing it. And in fact, that you need the diffusion of the inhibitor to be larger than the diffusion of the activator. So in that sense, you have some way of fitting these parameters. But the idea of trying to look at it geometrically was really to try and again find the relevant parameters in these systems, where the actual system is sort of more noisy. It's it's not homogeneous. It's and so is there some way that the geometry tells you? Again, it doesn't tell you how to go from the microscopics to the phenomenon, but does it tell you what sorts of parameters are relevant to fit given an experiment? And what I'm going to go to is to say that actually writing the equations. So here I'm starting with the model, but writing the equations in the unstable manifold of this model actually generates for you equations in 2 dimensions. The unstable manifold here was 2-dimensional, which are actually in a sense universal, and universal in the sense of Landau theory, that they're really they're only you'll always get something which is of the form r dot equals alpha r minus beta r cubed, and then that is sort of enough to capture the important dynamics, which is that you have some you have 7 fixed points. So this is actually with 7 cells. Here there was 5 cells, so you have some 5 different stable fixed points. So here you'll have 7 different stable fixed points, and I'm only showing you one part of the whole whole 2 pi angle. So I'm only showing you a bit of the angular direction, and the radial coordinate in the unstable manifold has is actually quite simple. And so fitting these parameters, I would argue, really captures the sort of whatever there is that's universal in the system, in the sense that whatever there is that doesn't vary given the noise and given the other fluctuations in the parameters that you could have. So I'll show an example of this. I'm a little low on time, but one can now, so just to go quickly, one can go to the continuous model. Again, this was a discrete case. In the continuous model, again, the crucial insight in Turing systems is that actually the unstable manifold in all of them is quite low dimensional. And in the continuous system, the angular behavior kind of becomes trivial. You just get, you know, a infinite set of fixed points on the boundary, and so you can take, for example, two radial directions. You can sort of change parameters. So you have you have a four-dimensional unstable manifold, and again, it turns out that the equations in that in those two different unstable modes are universal. And again, universal I'm using here a little loosely, but you can understand it in the sense of Landau theory. So that allows you, so here in blue, I'm showing you the flow lines from my potential. In red, I'm showing you the projection of an actual Turing system projected onto the unstable manifold. You know, there are several consequences of this. You know, the angles lock together, and you can sort of verify that um, uh, again in the actual dynamics versus the simulations. And you can really look at the full model behavior and compare it. And so somewhat, you know, non-trivial behavior of the dynamics can be captured quite simply using, again, this is sort of an infinite dimensional system, which I'm just modeling using these two radial variables. It can do quite a good job um, and you have to map, you need some way of mapping the unstable manifold to the actual activator variable, which I won't go into the details of, but it can do quite a good job of modeling the dynamic behavior of what's happening in this Turing system. And so, I mean, how is this sort of relevant is, I think, goes back to the question where, you know, in general, you have some system which shows some pattern state. For example, here I've taken a two spatial dimensional variable, which shows the appearance of some spots. And these spots are sort of inhomogeneous. They're not quite symmetrically placed, as you can see. But when you project this onto you know, the correct variables, then you see that the parameters that I had in these equations that I wrote down, they vary for these different spots. But that variation is really what you want to capture in a fit to this kind of system. And one would argue that here, the kind of system I've taken, I put in noise in all the parameters. I put in some you know, some crazy function, which is, you know, which is the kind of thing you would expect in the biology. You wouldn't expect some stably patterned spots, but rather you would expect some small inhomogeneities and so on. And these equations, I think, more accurately capture what is universal in this behavior. So actually, I think I'll skip over this final example of you know, bristle patterning in Drosophila, where again you have a system where you know, you have yeah. So I won't go into the detail of this because I think I'm running out of time.
You can take a couple of minutes if you want.
Okay, fine. So maybe I'll just quickly go through it. Where this is this is in Drosophila, and it's in the back of the fly. You get these sensory organs, and this is at some stage after pupa formation. You image, and then you see these are fluorescent markers, and these dots are sort of sensory organ precursors, which are on a layer of epithelium. So these are the sensory bristles that form at the back of the fly, and you're imaging this at a certain stage of development. And again, you have some, you know, it goes through again this notch delta signaling pathway, and the geometrical model for this again would be to say, well, there are two possible fates you can take at this stage. Either you get a sensory organ or you get a epithelium, which is shown in green, and you want to see how do you get these different spots on the back of the fly. And so if you take just one of these layers, one of these vertical layers, you can write down a pretty simple model of this, which is just one dimensional, which is just giving you these two possible fates. What that does is, again, some of your cells, so here I've taken 8 cells, some of them are going to become sensory organs, some of them are going to become epithelial, and some of this, so you can say you put in some parameters in the model that have some of them go to one and most of them go to the other fate. And so again, this was a geometrical model which was, you know, which is sort of nice, and it turns out you can write down this model also as a potential plus a metric. So this is sort of just another example of more of the same, where there's this original model and you can capture this original model with a potential plus metric. And here again, the geometry actually tells you that here the unstable manifold is not low dimensional. And the way it happens is you go to some saddle and then you actually get a starburst of saddle points, and then slowly each of the cells decide what they want to become. And also, by the way, the difference between these two is sort of irrelevant given the kind of data that's available. So, one could try and get closer agreement, but it's not, that kind of data is not available right now. And I don't think will be for some time. So this is just to say that, let me conclude by making just a small, I guess, philosophical divergence, but which I think has been theme of many of the questions that came in so far. But universality in physics, I think one understands quite well through, of course, the renormalization group, through, one has some systematic way to talk about how microscopic systems lead to, you can have a low dimensional parameterization of them by coarse graining and by removing irrelevant degrees of freedom. And I think universality in biology is sort of much harder to talk about, and it's certainly quite hard to have examples where you can have a coarse, have a proper coarse graining procedure. But I've tried to show here that um, imposing constraints on the dynamics, like genericity, and using things like, well, simple things like bifurcation theory and Landau theory, sort of gets you quite far, in the sense that it allows you to classify models of different biological systems that are being made, and which are being very particularly modeled, because they, people try to look at the particular genes involved, and you can really collapse them onto much more low dimensional systems, which are also more principled parameterization of the data. So with that, I guess I'll skip over the future work, and maybe I'll just conclude and say that Waddington's metaphor can be converted into a mathematical statement. It's possible to classify models of self-weight specification in low dimensions into these normal forms, phase diagrams, and that these potential models, as I said, are a principled way to fit data. And Turing pattern formations is the classic example in developmental biology, can also be written as a potential. So I'll conclude. Thank you very much.
Thanks, Archishman. Wonderful talk. Questions, comments?
I have a question. It's the following. It's maybe a very stupid question, but you see the way you have modeled the system in terms of low dimensional parameters is that, okay, a certain thing has to happen in nature. Actually, the organism becomes this or that. So you have some idea of the fixed points, and perhaps by varying parameters, you know the qualitative behavior around the fixed points. But what is fascinating here is that there is this whole microscopic theory in which there are all types of concentrations of different chemicals, linkages, etc. Is it that in some sense evolution designs those things in the microscopic theory to get to these endpoints? Just so fascinating stuff that you talked about, and I just wanted to ask this question: whether there, what is the reason why the coefficients of those microscopic equations are what they are?
Right.
Is it to somehow arrive at something like this, that nature practiced a lot and arrived at this concentration so that you get this type of final product? Just some comments if you have.
Actually, I mean, that's a very interesting question, because in fact, Waddington was interested in studying evolution, and his whole idea of a landscape, the kinds of experiments he did, he wanted to make statements about evolution. And what he wanted to say was that random mutations are actually not the relevant thing that one should be just thinking about. It's not just random mutations in natural selection, but there's also this landscape which directs you, which is canalized, and directs you into these particular fates. And so this map from genotype to phenotype is a very particular map which has been evolved over several generations, and that map sort of makes the search problem quite different from simply a random mutation problem. And so in that sense, I think that he was certainly thinking himself about evolution. But maybe a little closer to your question, I think this is to do with the challenge of actually finding a coarse graining in these systems, because these are evolved systems. And I think one is used to, in physics, one is used to homogeneous or random systems, and both one knows how to deal with. but these are in a sense somewhat specific. These are systems that have been evolved for some particular because they've seen some particular environments in the past. And that means that your small perturbations in the system could either have no effect, local perturbations, or they could actually mess up the whole system, which is again something that's not typical, I would say, in physical models. And yeah, so whether so canalization in particular, and the fact that you have this low dimensional thing, Waddington was tracing to the fact that when the environment changes, it's sort of one strategy to deal with fluctuating environments. When the environment changes, one strategy that an organism can have is you still show the same phenotype. Because there are other strategies, for example, the immune system just mutates very fast and deals with the new environment. So he was definitely tying the fact that they are low dimensional to evolution. And I think there are a host of interesting different questions there, which one has to deal with.
Can I make a comment at this point?
Yes, please.
Vijay?
Yeah, please go ahead.
Yeah, this is just a point of clarification with regard to Spenta's question. Waddington was very much a conventional Darwinian selectionist. So I think the point is not so much about whether evolution by natural selection led to these rather complex structures that we have today, but rather whether there were levels of control, if you wish, or levels of organization above the gene level, which also played meaningful roles. And his metaphor of this developmental landscape, which he thought might also stand for an evolutionary landscape, was to indicate that over and above the genetic level, there are levels of interaction, mutual interaction and mutual influence, let's say at the level of cells, which might reduce, to go back to Archishman's language, might reduce the huge dimensionality of the intracellular landscape into a much lower dimensionality of an intercellular landscape. This very roughly was the way Waddington looked at things.
Thanks, Vidya. Krishnan?
Actually, what I wanted to ask was something along the lines of what Nandita just told, which was whether, following from Spencer's question, whether some of these microscopics do end up in things like control modules to guide the kind of changes in the geometry that we want to perhaps see in order to land in one developmental fate or the other. So, no, that was, I think, but that was already kind of what mentioned.
Mm-hmm. Mm-hmm.
Okay.
No, yeah, I would agree with that. I think, that there's definitely some level of control that is very important in thinking about these systems and the way it's directing. In that sense, control theory is a relevant mathematics to talk about it.
Someone named SGP, could you please, I don't know your name.
Yeah. Hi, Archishman. Yeah, I had a question about the complex system that you started out with. If you sort of find the slow modes in that system, the slowly evolving modes, and identify the dimension of that, would that correspond to some of the dimension of the slow mode corresponds to the extremely low dimensional version that you talked about? Would it reduce to that? And also other ways by which you can convert this low dimensional models and then map them to high dimensional ones? Like, so you said it's projection, right? Other ways by which you can extract that projected information from these models?
So I don't know if I fully understood your second question. The first one is, that's part of it, certainly. That one could look at the time scales and you could do some sort of coarse graining in time scales to get a lower dimensional version of these complex models, but that's not all of it. I think it also does depend, the way these networks are structured. And the structure of the networks is not completely, you can't just look at the time scales and do the coarse graining, I think, though that does get you some of it, some of the dimensional reduction. the second, I don't know I fully understood. You're saying go get back the information you lost, or
Yeah. In the sense, are there ways by which you can couple these systems and then reproduce some of the complex behaviors that you would see in the macroscopic system? I mean, are there rules or constraints that these would impose?
On the microscope. I guess they would, but I'm not sure if I know what. I mean, sort of how one would write that down. But certainly, I guess you could impose the same constraints on a big network model and try and see what subclass of models one gets, and whether there's anything interesting there. Yeah.
Okay. Thanks.
Rajesh?
Hi. Yeah, thanks for the nice talk. I'm not sure the question I'm about to ask makes perfect sense, but I was just wondering about this low dimensional reduction of these Turing-like systems that you considered. You starting with the continuum model, you have essentially only a couple of really relevant degrees of freedom. Is this something like in height? I mean, these equations are, does it make sense to think of these equations roughly as some kind of dissipative hydrodynamic equations? And if so, then is this low dimensional reduction something like these lowest quasi-normal modes or lowest sort of there's typically one mode which is the sort of the dominant dissipative mode, or the so is that a reasonable way to think of these things? Or?
Yeah. No, that's a reason. I mean, that's a pretty accurate analogy. I mean, this is these low dimensional manifolds are just sort of the most unstable modes in this, which are at non-zero wave vector in these systems.
Okay. So that's essentially what these radial modes and the ones that you looked at are. Okay, great.
Right.
Before I go to Krishnan, I had a follow-up question. So I think similar lines. So this is the amplitude equation formalism that one writes for patterns. I guess this is very similar to that.
Right.
And the equations for the amplitude equations are basically Ginsburg-Landau type concepts.
Right. Yeah, I mean, it's not quite the amplitude, because usually, I mean, one still includes some space in the amplitude equation.
Yes.
But yeah, certainly there's
the zero wave number.
Yeah.
Amplitude.
Sorry.
Krishnan has a separate question.
Right. So I was thinking, sort of in the just a slightly more broader context. So as far as I understand, during development of organisms, it's not just that you have to end up, let's say, with the correct proportion or a correct pattern of these different cells. The time axis, by which I mean the sequence of events that follows, sometimes also is important. So you want to have cell fate 1 and 2 in the early stages, and then cell fate 3, 4, 5 in the later stages after a certain time. Now I was wondering, so this kind of formulation definitely helps sort of geometrically, without going into the microscopics, capture the decisions between 1 and 2. I wonder if something can be done about the time of these events as well.
Yeah, no, I think that's a very interesting question. And one thing is that one could link up different developmental decisions by linking up these low dimensional landscapes and using the hierarchy, so to say. And so things that happen at different times, one has one decision, and then one links that to another landscape where there's another decision, so on. So that's one thing. But I think what you're also addressing is something that hasn't been done, where, for example, in C. elegans, you can have these mutations that mess up the timing, heterochronic mutations. And how one would parameterize things like that, I think, is an open question. But certainly it's it's an interesting one.
Vikash?
Yeah, so I mean, it might be very naive, but this landscape metaphor is also used in proteins. So I was just like wondering whether a similar kind of an approach could be there, and how do we like make analogous kind of statements about like a protein when it is like having a particular like, when it's actually going through a structure, like forming a particular structure from the DNA. So if you
Right,
could
right,
comment
right.
on about.
Yeah, no, it's a good question, and I don't. I think, I mean, it's worth investigating. I mean, the other thing in proteins landscapes also, you actually want thermodynamics, and so it's a little here. I'm not at all dealing with you know, the noise is sort of trivial. It's not really doing much except permuting different cell types that you're taking. But yeah, I mean, proteins I actually don't have too much to say about. So maybe just taking advantage of your question, I can also say that the actual development also happens. I mean, if you think about it, in, you know, there's some morphogenesis, there's some mechanical forces, and there you have a much more natural idea of a landscape and of potentials. There are forces, you know, that correspond to potentials in the usual physics sense, and you know, this landscape doesn't quite, well, it's quite unclear what it has to do with that landscape, and whether there's something that combines the two, and what that would even look like. But proteins, like, I mean, it's an interesting question, and I think worth thinking about, but I don't have much that's useful to say.
Any other questions, comments? Okay, so if not, let's thank Archishman digitally. Thanks, Archishman, for a wonderful talk. We all enjoyed.
Thank you.
Thanks, everyone. Thanks for coming. Take care and stay safe.